All right, students. The learning targets for this activity today. So hopefully once we are done, you will be able to apply your knowledge of chromosomes that you even already have to this simulated chromosome. So we're going to find things like telomere and centromere, genes, both introns and exons. Introns are those non-coding regions that you learned about in the DNA profiling. Exons are those coding regions that code for a trait. Remember, EX for expressed. Those are going to be expressed by your genes for like hair color and eye color. Our P arms and our Q arms. We're also going to find the promoter gene as well. So I should have thrown that in there. Our promoter and our terminator. You are also going to examine how regions of DNA are really determined by their DNA base pairs. So DNA is spelled in those four letters as A, C's, G's, and T's. And based on how they're put together, determines the traits and determines the gene in the chromosome. Much like our alphabet is 26 letters, and however we arrange those letters make words, the DNA is in predictable stretches as well. And then we are going to start laying the foundation for what we're going to learn a little bit later next week of translation of a protein code from a protein. So we're going to start learning how to read the blueprint as if we were architects reading a blueprint to build a house or a car or whatever it might be. Now, we are also, just to kind of review some of the things we already know already, we know that DNA is a series of these, A, C's, G's, and T's. Remember, C's partner is G because cars are in garages and apples grow in trees. And so we know the code there. We've talked about the anatomy of telomeres and the short and long arm, the centromere that the spindle fibers attach to whenever this gets pulled apart during cell division. And we've also talked about the gene that has a promoter. Now, sometimes there is a thing called an operator. That's not in our activity today. It's a little simpler than a real gene, but that promoter is going to be the beginning of the gene where some things are going to attach to start making copies of this gene. Then we have exons and introns, and then we're going to have this thing down here at the end of a terminator or a stop gene segment. So that kind of review that. And so what I'll ask you to do is every one of these slides, there's going to be a big video for like task 1 and task 2. This likely will take you a couple days. So what I would ask you to do is work for 40 minutes. Or if you're at home, maybe get this done and you won't have anything to do tomorrow. So, but my anticipation is it'll take somewhere between probably 60 to 80 minutes to complete this whole thing. It will depend on how quickly you are. It may also depend if you are working along with someone. So unfortunately, our students at home won't be able to work with anyone unless you have somebody at home you are working with, or if another biology student wants to come over, you could work with them, I suppose. But the folks here at school, I will probably encourage you to work in teams of 2 just to help save resources, although this can also be done individually, no problem at all. All right, so that's what I have to say about this little intro part 2. Now you'll move on to the next task. Read that. There's usually going to be 2 slides per task. One's going to be video, then there's going to be a set of things you have to be inputting or information you'll need to solve the task. But hopefully I've organized it well and use the videos wisely, and thanks for watching. See you next time.